Hello, my name is Jun, and this is my final project presentation on Camelyon16, and I worked alone on the project. And so, to begin with, let me walk you through the code that I wrote. And so, in the code, the I first start with all the trivial, bringing in all the importing, downloading, installing, mounting Google Drive, and extracting the data sets using OpenSlide shown in class. And then I demonstrate by plotting three slides that I bring in to demonstrate that I can really use them using OpenSlide. And then I create the find patches from slide method, which takes in a slide, original slide, and then breaks down the image into 256 by 256 pixel into many number of tiles or patches, whatever you call them, and then uh, this method takes in the original image, breaks it up, and then produces binary image using Otis thresholding method, and then compares the binary with the binary patch with the original patch and decide whether uh, the patch contains tumor or not. And it outputs slide path, tile number, and tile index, the location relative to the original slide, and then whether each patch includes a tumor. And then I find all the tumor cells, and then this method automatically throws out the non-tissue parts. As you can see, in this area, it would be considered useless in training. And then I extract the tissue, separate the tissues with tumor and non-tumor by using the Boolean, and filter out in the data frame, and then I append it all together into one huge the data frame, and then you see at the end all appended together. I have 16,000 patches, tiles with tumor, and almost 29,000 tiles or patches without tumor. And then I move on to extracting 10,000 because I'm using Colab, and Colab has a limited processing power, and then it's very prone to disconnecting. So during training, and I just had a hard time training for a long period of time consecutively because yeah, has to depend on internet connections also. So with all that, I extract 10,000 images samples from tumor and non-tumor samples, and then I shuffle them, and then you see. And another thing why I emphasize 5 to 5 ratio is because I do another experiment with a 5 to 3 ratio, where the 5 is the patches with tumor and 3 is patches with non-tumor, and I'll talk about the results later. But yeah, that's why I emphasize the ratios of the training data. Because initially I thought if you had more samples with tumor patches, or if you had more number of tumor patches than the number of non-tumor patches, then I thought the model can concentrate more on the features of the tumors. But the result says it doesn't, and I'll talk about that later in detail. And I generate data, training data set. So in this method, gen images, I just I get I break up the I get the tile location, like, which is the content with the slide, and then I extract the specific index of an image patch out of there. And this so in all this I use the in my entire training data set or test data set, validation data set, I generate the patches from non-sampled, non-downsampled, so 1 to 1 ratio original image, the highest resolution, because in the original paper that won the contest says that if you use the highest resolution, images, like the result doesn't really matter whether you train on different multiple levels of zooming. So yeah, I took the advice and I create the generator for the image, and then I create this small miniature VGG16, which is CNN. As you can see, it contains convolution 2D layer, max pooling, convolution, dropouts, and convolution layers. So this is a base and has an increasing number of filters. So 100, 200, 300, 400. And this is a miniature version of VGG16 because I also ran this experiment on applying transfer learning on the VGG16 model, trained on ImageNet dataset, but result wasn't really good. I'll talk about that later, but it's just code run through. And I also tried the exactly same model with different activation, because I read on a forum, one of the forums, that it worked better if you use ELU, exponential linear unit, instead of ReLU, rectified linear unit. So I'll talk about the result later, but this is the model. And then I split the data sets into training, validation, and test in the ratio of 80%, 15%, and 5%. And I did not do any augmentation on the training data, except for I did shuffle, but I didn't do any physical transformation like rotational, zooming, horizontal, vertical, because I already had enough number of training data set to make a validation, because I had a hard time just working with Colab with its memory connection issue, so I want to just try to make it as compact. And this is just a shuffling of the data set, splitting, and I have 16,000 training samples, 3,000 validation samples, and 1,000 test sample. Then I train the model, and you can see the results here, and I also plot the result here. And then I sample 32 images from the test sample. And also in all my shuffling, I use the random states, the same for other experiments that I did. And so just to make the same data set the same, equal, unbiased. And here's it's a image of 32 samples, example of 32 samples from a training data set. And yeah, and these two methods help me plot the heat map and the predicted heat map, and just adding them together. So here's the result of original patch with truth or the mask patch, and this is predicted from my model and the predicted mask, and this is a heat map overlaid with the original image. And you can see this is a hinting, very red on the top, hinting that this is cancer. So it's kind of doing its job, but not on the bottom. And I also do the histogram of patch pixels, each pixels, and then see what's the probability of them, and count the number. So there are a lot of there's a lot of confidence in this prediction, you can say, because there's more a lot of counts dominating the positive 100% sure that this is tumor patch. So it's with confidence. And second, third, 4, 5, and 6, I have 6 results. And then at the end, also have another metric system to see whether my model is trained well. And use a confusion matrix over the entire training data set. As you can see, using the test samples, I create a generator, image test set generator, and then just do the confusion, and you can see the accuracy. So other experiments, other experiments that I did with a different ratio, 5 to 5, 5 to 3, and using transfer learning on VGG16 and using ELU activation, I have a similar format and same code, similar code used throughout. And so let me talk about the result. So just to just go over the data generation again, I test with two different cases. So whether the first case is a tumor with 5, ratio 5, and patches without tumor is 3. So I use 10,000 tumor images, patches, and 6,000 non-tumor patches. And I didn't do any augmentation except for shuffling because I had enough training data set for my collab to handle. So first result I'll discuss is the ReLU. So my minimal miniature VGG that I implemented, and this one has a 5:3 ratio of training data set, tumor to no tumor, and this one has a 5:5. And you can see the values here. And at the end, my validation accuracy was similar, very similar. But if you look at the loss functions, you can see the in the 5:5 fair unbiased data set, it decreased faster and reached higher accuracy faster. And you can really see the difference when you go into the heat maps and the histogram of the pixel predictions. Even though the two comparing samples are not the same because the number of the uh, entire training data set was different. The ReLU 5:3 ratio has a less number of training data set, but I don't think it really mattered. And you can see heat map wise, you can't really see. You can see 5:3 is also performing well, but once you go into the histogram, you can see the 5:5 model is predicting with confidence, where also there's little high risk on confident counts. And you can see that's more pronounced as you see in the other examples. So example 2, 3, and 4. You can see in the patches, you can't really tell the performance by the eye. You can see it's performing better for the 5:5, but you can really see the details of how it's performing in the histogram. Look at all this unstable, like unsure, unconfident predictions made by the model. And whereas it's very low here, very high, very high. So I concluded in my experiment that uh, unbiased, the 5:5 data set performed better, and later on I use 5:5 for everything. And then now I wondered what kind of a model would perform better. So I tested with a smaller miniature version of VGG16, and then I wondered what would the actual performance be compared to the VGG16 transfer applied with transfer learning on image net dataset. And you can see the training accuracy is very low, and the training loss is very high for the VGG16. But you can see the validation accuracy is pretty low. So you can't really tell from these validation accuracy. But you can see in the confusion matrix result, you can see that the miniature version of ReLU or the VGG16 that I implemented worked better. And yeah, and this is a transfer learning, so I didn't have I only had 2 convolution layers that I could just fix the parameters of. And you can see VGG16 doesn't perform well at all when it's on the transfer learning. Look at this bell-shaped curve of the probability, because this tumor is basically yeah, it's beyond 0 0.5. The majority is beyond 0 0.5. That's why it's detecting non-tumor, but that is wrong. There's no yeah, gradation. There's no difference in level. Maybe I suspect this is due to the just few two convolution layers that are just getting fine-tuned, and uh yeah, I think that was the issue. Because as you can see, they all have like similar bell-shaped curve. They're not identical, but they all have, and then doesn't really predict well. And I'll just quickly show you guys the VGG16 model summary, just to clarify some things. As you can see, I only have two convolutions, and then I limit the VGG16 so it can't train on itself, so trainable into false. So I just fine tune it, but the result seems really bad, so I throw out the VGG16, and then I try to test it with ELU activation instead of ReLU. So it's exactly the same model, but instead it's getting activated by ELU, as you can see. So it has the same unbiased ratio of in the data set, and you from the values perspective, you can see the validation loss dropped faster actually for ELU. So you can say from this perspective, ELU trained faster, or reached the, I guess, optimal va validation loss faster first. And you can see, but for training accuracy, uh, wasn't really the case. But for validation of for both validation cases, the accuracy reached faster for ELU and loss dropped faster. So from this result, ELU actually does seem to be working better. But let's look at the heat maps and the histogram to see which model performs better. So from the heat map predicted mask, you can't really tell whether which one's working better, and it's very hard to tell from the prediction histogram. Uh, you can see they're all pretty much the same. Some images ELU performs better, some images ReLU performs better. In this case, like ReLU performs better, but some this case ELU performs better, and honestly, this is not enough to result to definitively say which activation works better. But from my perspective, my guess is that ELU would have more. ELU has a more higher loss function, so maybe you can minimize it more. But to be honest, I think it's inconclusive. So in my case, you can reach both accuracy of 97.8%, 97.6%, which is almost the same, and just using the shortened version of VGG16 instead of the transfer learning. And yeah, that is the final result I got for the project. Thank you.